interested in this area because I've got a slightly weird background. So I trained in medicine a very long time ago. I then worked as an academic neuroscientist at Oxford, then at Newcastle University. And at a sort of relatively mature age, I sort of stopped being an academic and I moved into sort of consulting and then into finance. And I've sort of oscillated back and forth between sort of consulting, finance, and also bits and pieces of real biotech, including some biotech drug discovery. And I think that rather peculiar, so I'm just going to try and forward my slides, that rather sort of peculiar background has given me an interest in R&D productivity. And I'm going to kind of give you one of the big messages right up front, right, to sort of cue you about what I'm going to be talking about. And I think there's quite a good analogy for drug R&D or drug discovery. And it's that finding drugs is a bit like finding tiny oases of safety and efficacy in vast biological deserts, right? So nearly every biological or chemical opportunity you look at probably ain't going to work, but a few do. And the screening and disease models in which we decide whether a drug target is a good one or whether a compound is a good one, they are the things we use to try and point us towards oases, right? But they only do that if you give them a bunch of candidates and they score them in the same way that a human would, right? If a sick person took the same thing. And if they don't, they're going to head in the wrong direction. And in particular, I think, possibly less so in recent years, but certainly there's a period probably from the 80s until the early 2000s when sort of brute force efficiency took over. And that was a bit like running around the desert at great speed with a very bad compass, right? So it gave the impression of progress, but actually very often you were heading in the wrong direction. And that's because arguably the screening and disease models weren't good enough. So I'm going to start with a caricature, but this is the drug industry's caricature, not mine. And it's how the drug industry says, you know, when talking to investment analysts like I used to be, or talking to politicians, how drugs are discovered. You know, you have something called target identification. I'm going to move a cursor here, and Lisa, can you see me moving the cursor? Or
yes,
anyone?
fine.
Yeah, fine, fantastic. Okay, so you start, at least in the caricature process, by doing something called identifying a target. And targets are pieces of biological machinery that are presumed to have gone wrong in a particular disease, right? Or if it's a bacterial infection, it might be something that's gone right for the bacteria in a particular disease. A lot of this comes from, for example, looking at genetic associations with disease risk. You know, so you might find that a particular protein in certain people is different to other people, and maybe that is related to the difference in disease risk that people get. There's then a process called target validation. You know, so for example, in this case, you know, you might express the gene that you've identified in people in a rat, see if the rat get a disease that's a bit like the human disease. And if they do, you'll presume the target to be validated. What you then do is you then develop a method for trying to test therapeutic interventions, often small molecule drugs, but sometimes other things, to see the extent to which they bind to and modulate the behavior of the target. You then test lots and lots of things against the target to find things that seem to do a good job in modifying its behavior in an appropriate way. You then do something called lead optimization. So let's suppose you're trying to develop a headache drug and you've got something that really, really binds your headache target, but it can only get into the skull via an intracranial injection, right? Well, that's probably not going to be commercially successful. So you'll try and do a bunch of chemistry so that people can take it as a pill and other things as well. You then see if it cures headaches in rats and, you know, passes a whole bunch of safety related hurdles. And if all that happens, then you can start your clinical trials in people. And this, again, it's a caricature of the industrial process, and lots of other things happen as well, and it's more diverse than this. But effectively, by building this kind of industrial process, you know, you've effectively industrialized a specific view and a specific aspiration, and the aspiration is something as follows, right? That actually there's lots of pieces of biological machinery which misbehave and cause a disease phenotype. You can drug them, that's in screening and lead optimization. And also, there still exist lots of targets and lots of drugs that may be tested against them. And if those assumptions are true, this is a great process for delivering at very low cost a large number of successful drug candidates into the clinic. Right. So that's the sort of industrial basis of, or at least a caricature of the industrial basis of drug discovery. Now, it became clear to me working in investment working in the commercial world in the very late 90s and early 2000s, that there was a big problem. Because all of the steps of the industrial process, all the technologies that underlie the steps of the industrial process, so for example, DNA sequencing, well, that's terribly important for target identification. X-ray crystallography, well, that's really important for thinking about screens and screening. Those sorts of activities, and a whole bunch of other ones as well, which I won't talk about, had got massively better over the last few decades. So this is a logarithmic axis, and this is a logarithmic axis. This graph is now out of date, but between the late 60s and 2010, DNA sequencing got 10 billion times cheaper in terms of unit cost. X-ray crystallography, that allows you to look at the structure of a protein, so you can kind of guess what drugs might bind it before you do any expensive wet science. Well, that got roughly 10,000 times cheaper over a similar period. And I could point to a large number of other technologies that are believed to be important for these individual steps. But over that period, the amount of money that the drug industry spent per drug discovered or drug approved by the FDA fell by roughly 100-fold, right? So, and this is real terms, so this is inflation adjusted. And I became inordinately interested in how you could have a process where all of the inputs get hundreds, thousands, millions, or billions of times cheaper. And these are the inputs that most people doing the process think are important. But the process as a whole gets 100 times more expensive. Now, things actually have improved a bit since 2010, and that's not going to be a primary part of my talk. I may allude to it, but again, if anyone's interested and I forget to say anything about it, prompt me at the end. But for me, the sort of overwhelmingly interesting bit is this, right? 100 times more expensive in terms of drugs approved per $1 billion spent by the global biopharmaceutical industry in the face of massively improving input efficiencies. And I use this slide to remind the audience that there are really two ways to explain something like this, right? So, and I'll tell you what the two pictures are first. The picture on the left is a guy sitting with something that's called a Tasmanian tiger, or sometimes a Tasmanian wolf. And on the right, it's not actually an alchemist, but it's a picture I got that looks like an alchemist. And let's imagine that this guy's trying to transmute lead into gold, right? So if your inputs have got better and faster and cheaper, and your outputs have got 100 times more expensive, well, you could be a bit like this guy sitting here, where actually it doesn't matter if your Tasmanian tiger technology gets thousands of times better, because the last Tasmanian tiger died in Hobart Zoo sometime in the 1930s, right? So there aren't any of them left. So there's a kind of resource depletion set of explanations. But then there's another logically possible set of explanations, which is the qualitative nature of things that people did in the 1950s, 60s, and 70s were different from the qualitative nature of the things that people have done in the 90s and 2000s. And actually what we've done is we've progressively replaced more effective search methods with search methods that have much lower unit costs and much higher brute force efficiency, but actually are much worse, right? So again, we're trying to transmute lead into gold, something we can't do. And again, as I generally remind folks in the drug industry whenever I show them this slide, the more you disagree with one of these explanations, unfortunately, the more you're forced to agree with the other. So that if you think actually there's lots of fantastic opportunities out there, then you're forced to conclude that actually we somehow must be doing R&D in a much less effective way. On the other hand, if you think actually we're really doing it very well and we're incredibly effective, then you're forced to conclude that there's been a real sort of catastrophic or a potentially catastrophic depletion of opportunities. And I'm going to introduce a slightly geeky decision theoretic slide, which is going to formalize that a little bit, and I may come back, because I will come back and talk about some of these ideas, but there's sort of two. So if I think about this previous slide, right, here's, okay, we've run out of things, or there isn't anything to discover, here's, we're doing it wrong. You can think about that a bit more formally in terms of finding drugs for diseases. So, we can imagine that the screening and disease models we have are effectively the tools we have for deciding whether therapeutic candidates are good or bad, but also the sort of chemical technologies or the antibody technologies, they're ways of making therapeutic candidates. And if you think about the diseases which may exist, well, some of them are probably not pharmacologically tractable, but maybe modelable, right? So, for example, if I would guess traumatic limb amputation, that is pretty modelable in an animal. It might be sort of grisly and unpleasant to do, but I'm sure that the pathophysiology of traumatic limb amputation in some animals is pretty similar to people. But no one would expect one to be able to discover a drug that would suddenly make a limb grow back in a higher mammal, right? So that's something that may be modelable but not pharmaceutically tractable. And then there may be diseases that are pharmaceutically tractable or pharmacologically tractable but not modelable. And I would argue, actually, history suggests that human depression may be such a disease. So arguably all new classes of antidepressant drugs were discovered through observation in people, right? So clearly there's chemistry that helps with depression, but actually it's very hard to find that chemistry by testing it in models. So you can imagine that if you've got an R&D process, you've got a bunch of notionally approvable compounds that might exist or might be able to be created. You've got unapprovable candidates. And then what you do is you go through a decision process where you use your models. You've got a sort of true positive rate for detecting good candidates and a false positive rate, where you've got at each step in the process, by finding in your initial screen, you find lots of the good candidates and you reject lots of the bad candidates. When you get to the next step of the process, you progressively enrich Q, which is the ratio of good to bad candidates, right? So we have a sort of pharmacological tractability, i.e., what proportion of randomly selected compounds will work in the disease, and then we've got a bunch of decisions that we make to try and enrich the compounds progressively through the R&D process, right, before we put them into expensive human trials. So, following that bit of formalism, I'm going to try and give a sort of short summary of what I think the main drivers of Room's law are. So this is the progressive, the fact that it's so much more difficult to discover drugs now than it was, despite the fact that technologies have got better. And I think the primary problem is something that I described in a paper I wrote back in 2012. We called it the better than the Beatles problem, which shows our age and lack of fashionability. But the analogy is this: imagine that any new music that was launched had to be A, better than the Beatles, B, you could download all of the Beatles back catalogue for free, and C, you never got bored of listening to Beatles. Right? If you had those three criteria, it would be very, very difficult to launch new music. Now, the drug industry is effectively an intellectual property business, and it has exactly those better than the Beatle characteristics, because what happens is drugs are launched, some of them turn out to be very good. Most of those then eventually become generic. And what that does is it effectively makes it difficult to compete with new drugs in the same therapy area. And what the graph on the left shows, it shows the proportion of US prescriptions that are for generic drugs. So, in 1994, most drugs prescribed in the US were branded. Now, 90% of drugs prescribed in the US are generics. So you've got this ever-improving back catalogue of very cheap medicines. And what that's done is it's pushed the residual R&D into those areas where historically the drug industry has been unsuccessful, right? And if the drug industry has historically been unsuccessful in those areas for the last 100 years, which is roughly how long the modern drug industry has existed, they're probably difficult for one reason or another. Now, this is where I'm going to turn a bit more to disease models for reasons that I'll describe later, I think the Eroom's law and the Beatles problem point to the importance of disease models in R&D productivity. Because some disease models, if I go back to this slide, they're good at distinguishing between good and bad therapeutic candidates, right? Well, what happens to the disease models that are good at distinguishing between good and bad therapeutic candidates? Well, they give us good drugs, right? So what happens is the best in the Beatles problem applies to those diseases which are eminently modelable, where we have good preclinical models, right? And also probably they apply to the most pharmacologically tractable diseases, right? So we have this selective retirement of the diseases with the good preclinical models. And what we're left with is the diseases where the models don't identify the chemistry that cures people. And ironically, because we're left with the diseases where the models don't identify the chemistry that cures people, very often we keep using those same models for year after year. I don't want to pretend that's all that's going on. There's a lot of other important factors as well. I mean, the regulatory environment has changed massively over the decades. I mean, that's very, very obvious. But I think the decision theory stuff I'm going to show in a bit points towards disease models being an important part of the story, and also possibly an important part of the cure. Okay, so now I'm going to try and talk about screening disease models a little bit more formally. So imagine that we had infinite money and no ethics, right? We could, in principle, test a very large number of therapeutic candidates, and these could be drug compounds or they could be targets. We could test them in an animal efficacy model, right? And we could test them all in people in phase 3 trials. And imagine what this graph plots is the correlation you would get between the animal efficacy score and the clinical utility in patients. Or we could do the same thing in an in vitro test, some sort of test tube test of how well a drug might work. And again, we could think about how well it works in patients. So although these numbers are never actually measured in practice, right? If we didn't believe this in principle, if we didn't believe that doing better in our animal model was more likely to identify the red dots that work in people, or doing better in our in vitro model was more likely to identify the red dots that work in people, if we didn't believe that in principle, although we never practically measure it or rarely practically measure it, we wouldn't use the models in the first place, right? So the screening and disease models we use in R&D are the decision tools that allow us to suspect that some candidates are better than others and move them on in the R&D process. And these ones are just made up. Here are some real ones. And I think what these show is they show the sort of difficult data that biologists have to work with, right? So again, I'm going to tease some people on the, some of the views I sometimes hear people express, which is, why don't the drug industry just discover better drugs, right? Well, how hard can it be? Well, what these graphs show is actually it's pretty hard. So this is a graph where on the horizontal axis we have a measure of drug performance in this case, an in vitro toxicology model. So you don't need to worry exactly what this means, but basically drugs that are over this way look more poisonous, drugs up this way look safer. On this axis, this is a sort of ranking roughly of how dangerous the FDA thinks the drugs are in terms of liver injury. So 1 is actually the most dangerous. These are very risky drugs that have been withdrawn from the market or failed in late stage trials because they caused very severe liver injury. These drugs are very safe, right? The only way these drugs would injure your liver is if someone dropped a crate full of the drugs on top of you, right? And then you've got slightly more dangerous drugs in between. But what you can see is that with, circa 2017, the best in vitro liver tox model you could find, the correlation here really is pretty weak, right? So if we sort of set an arbitrary threshold and said we will decide anything lower than 0 is too dangerous to go ahead, well, actually what you do is you throw away quite a lot of relatively safe drugs and you still let through a lot of drugs that are very, very toxic, right? Here's another example. This is a screen, that I know a couple of people on this call, John, for example, would be familiar with this kind of thing. This is a screen of a class of drug called tyrosine kinase inhibitors. These are often used in cancer. And here's actually two different model measures, right? This measure is a very cheap measure that you can get at very high throughput. It's a binding affinity measure. So you would typically do this first in the R&D process. And then this measure is a more expensive measure, how much you inhibit the enzyme that you want to inhibit, more expensive, harder to do. And typically you would screen the compounds with this model before you put a subset of them into this model. And again, what you can see is, the correlation here is not great. There is some correlation, but, again, if you use this as a guide to that, you're going to get the wrong answer quite a lot of the time. And one can start putting, and in the work I've been doing recently, and again, I think there's probably at least one, perhaps two of my there's at least two of my collaborators on the call. You can try and start to formalize this a bit and put some numbers around it, right? So what these graphs represent, these are sort of idealized versions of what we saw on the previous slide. So these are effectively, you can think of these are probability density functions, where you can imagine we've got a universe of therapeutic candidates. We test them all on our decision tool, and then we test them all in clinical trials, right? And here we've got a good model, and I use the word model and decision tool to mean the same thing sometimes. Here we've got a lousy model, where the correlation between the score on the model and human clinical utility is weak. And what we can do is we can, get more or less stringent. We can say, well, we're only going to take really good things on the model. And we're looking for things that exceed some threshold of utility in people. And things that pass on the model and work in people, those are our true positives. Things that pass on the model but don't work in people, those are our false positives. And there's an important measure in drug R&D called positive predictive value, which is the proportion of things that you think are going to work that really work. And the reason that's important is the process gets more expensive and more ethically difficult as you go down the pipe, right? So by the time you get to human trials, you want to have a high confidence that the things you're going to put into people work, and high confidence that you've avoided false positives. And there is one little bit of mental maths or sort of a little bit of mental gymnastics one needs to do here, which is, let's suppose we want to use a really high selection threshold to try and improve the ratio of true positives to false positives. Well, to do that, you've got to test more candidates, right? So if you want to get the best one candidate out of 10, you have, or the best 10% of candidates, you have to test at least 10 candidates and find the best one. If you want the best 1% of candidates, you have to test 100 candidates. If you want the best 0.1% of candidates, you have to test 1,000, right? So throughput or brute force efficiency lets you search further out for things that score higher and higher on your decision tool or in your model, right? So let's see how positive predictive value, the ratio of true positives to true positive plus false positives, varies as you search further and further out on this axis using a good model and then using a bad model. Well, with the good model, if you start testing enough candidates, your true positive to false positive ratio becomes very high. Your positive predictive value can become quite high, i.e. the things that you think will work in people really do work in people. But if you've got a poor model where there isn't much correlation, the ratio of false positive to true positive stays low, even if you test lots and lots of candidates. So if we look at this graph over here, you can look at the shading is positive predictive value, and you can see how that varies with the predictive validity of your model, which is the degree of correlation between the score on the decision tool and clinical utility. And throughput, which effectively is how far out on this axis you search. And what you find is this, that for much of the parameter space that is relevant to drug R&D, changes in the correlation between your model and the human outcome of interest of 0.1 have a bigger effect on productivity than 10 or maybe even 100-fold change in your brute force efficiency. Right, so for example, when your model really doesn't correlate with human clinical utility, you do just as badly when you test 10 to the 7 compounds as you do when you test 200, right? And I'm going to give a good historical anecdote here, which I think illustrates the point very well. So the world's second useful antimicrobial drug was a drug called Prontosil or sulfanilamide. It was discovered by a guy called Gerhard Domach in 1932. And he was working for Bayer at the time. And in 1932, there really were not large libraries of drug-like compounds that people could test. And Domach had access to a couple of hundred compounds. And he tested a couple of hundred compounds, and he found this drug called Prontosil, which until penicillin came along was probably the most broadly useful antimicrobial drug there was. Between 1995 and 2005, the global drug industry, Glaxo, but a bunch of other companies as well, decided to throw genomics at the problem of antimicrobial discovery. And they had a very convincing plan. Their idea was to sequence the genomes of lots of bacteria, identify genes that were essential to bacterial survival, because if you could block the products of those genes, you might have a good antimicrobial drug. They then cleverly cross-referenced those against human genes to make sure that they didn't pick things that had a close homologue in man, so they wouldn't be toxic. And across several countries, companies, they ran well over 100 high-throughput screening campaigns against 70 or so targets, many involving more than 500,000 compounds tested. And they found precisely nothing that was worth putting into clinical trials in man. So there's a very obvious question, right? Well, how could a guy testing 200 compounds in 1932 find something useful, whereas people testing 10 to the 7 or possibly 10 to the 8 compound target pairs in 1995 to 2000 discover nothing useful? And the answer potentially falls out of the decision theoretic maths. If Domach's model had a very high correlation with human clinical utility, and the way that R&D was conducted later had a very low correlation with human clinical utility, you would expect one compound out of 200, the best one out of 200 in 1932, to be more likely to work in people than the best 1 out of 10 to the 7 in 2005. So why might Domach's model have been more correlated with clinical utility in man? Well, I think we now know the answer. Domach was testing his drugs in mice, which he had infected with bacteria and which had sepsis. And some animal infection models are pretty good. In 1995 to 2000, the drug companies were testing their compound libraries against isolated bacterial gene products in little pots. And we now know that decorrelated the results in the 1995 to 2000 period from human clinical utility in a number of big ways. So the first obvious source of decorrelation was that the compounds that drug companies have in their compound libraries are heavily enriched for compounds that don't get into whole bacterial cells, right? And they were doing the screening against isolated bacterial proteins. So that was one source of decorrelation. And then we also now know that the genes that are essential for bacteria in vitro cultures are not the same genes as are essential for bacterial survival in living things, right? So the compounds didn't get into bugs when you tested them in people, and the genes that you found in vitro weren't important anyway, right? So those two things meant that we decorrelated the model from the human outcome of interest. So the message from this part of the talk, and it gets a bit more general and applied next, is that the thing that nearly everyone already believes is important is more important than nearly everyone already believes, right? So no one in the drug industry thinks bad models are a good thing, right? No academic sets out thinking, I'm going to invent a really lousy model of disease X. But I think unless you run the decision theoretic maths, and also I think take a close look at the history, you underestimate the quantitative importance of very, very small changes in model validity. And I'll give you one more anecdote before going on to the practicalities. I spent quite a lot of my professional life working as a biological scientist. And if I plotted two graphs, one had a correlation of 0.6 and one had a correlation of 0.7, I would assume there's really no difference between those graphs, right? But actually, the difference between a model that correlates 0.6 and one that correlates 0.7 with a human outcome of interest could have the same productivity impact as doing 10 or 100 times as many experiments, right? So the quantitative power of small changes in validity is very, very high. So what are the practical implications? Well, and I think the first one is that certainly the training of biological scientists is blind to this stuff, right? So people aren't educated about how important model quality or predictive validity is. Another issue is that there isn't a common language to talk about these ideas, right? So, if you get a psychopharmacologist in a room with a pain biologist and a infectious disease discoverer, they will not have common language to talk about how good or bad models are, right? Another issue is that the statistics that people are taught as biological scientists are generally around hypothesis testing, and decision making is actually rather different to hypothesis testing, and the sort of statistics that it would be useful to educate people on if they want to know whether their models are giving them the right answer, they're actually very close to the statistics that you apply to diagnostic testing, right? It's things about ROC curves, true positive rates, false positive rates, false discovery rates, and so forth. And again, that sort of stuff is actually largely absent from conventional biological science education. Something I will talk a bit about is we should throw more effort at evaluating models. And sometimes when I say this to people in the drug industry, they think I'm sort of arguing for a council of perfection, right? i.e., look, how on earth can we evaluate models if at the moment there's no cures for disease X, and if we test a bunch of compounds in the model and then put it into trials, it's going to be 20 years before we know whether the model is any good or not, right? Well, that's not really what I'm suggesting. I think model choice is already... Oh, there's a good analogy here, which is what you might call Bayesian versus frequentist statistics, right? So there's a flavor of statistics called Bayesianism, about the sort of expectations of what will happen. And then you have so-called frequentist statistics, which is about counts of what actually did happen. And my view is that model choices at the moment are already Bayesian in flavor, right? So we use a model because we think it's going to tell us something useful. And all I'm arguing here is that we do some practical evaluation to try and drag our guesstimate of how good the model is a little bit closer to reality. And one very good way of doing this is to develop criteria against which you evaluate models before you have your model, right? Or rather, the thing to do isn't invent a model and then assert that it's a great model of a human disease, which I think is what happens a lot at the moment. It's to actually prospectively define what a good model of disease X would look like, and then as independently and rigorously as possible, evaluate your models against it. And this will help you decide how much to believe the results of a particular model, but also it lets you combine models, and I'll talk a little bit about that. A couple of other things is that I think at the moment, most of the financial analysis of pipelines is blind to model quality and decision quality. And I'm doing some work to develop alternative financial frameworks so that people can actually value better models. And then another thing, which I probably won't talk about much, is that there are also, I think, economic problems around investing in better models. So models have a lot of the characteristics of public goods, in that they make life better for lots of people who are trying to make money out of drug R&D, but in themselves, they don't earn very much money, right? And I think that means a lot of the investment in commercial drug R&D goes into novel chemistry, and investing in the models that would help us decide whether that chemistry was going to be useful or not is something that doesn't happen as much as it should. Right, so I'm just going to maybe make a couple more comments. Again, I want to leave plenty of time for questions, so I'm just going to touch on a couple of things. something that I've been doing lately is actually trying to get to grips with the practicalities of model evaluation. And again, there may be some of my collaborators on the call. If so, I'll say who you are at the end, but I'll thank you now. And effectively, this is, to a certain extent, it's about clever checklists, right? And for sort of mnemonic and ease of remembering reasons, I structure my clever checklists under 4 big headings. One is biological recapitulation, which is essentially a serious effort to articulate the degree to which the model captures the relevant aspects of the disease state. There's something around tests and endpoints, which is the model testing and scoring candidates in a way that's relevant to the human clinical state. There's a third one where there's actually a very large literature already, which I've sort of begged, borrowed, and stolen. And that's around the extent to which the model minimizes bias, noise, maximizes reproducibility, etc. And then there's another area of evaluation, which I think is very under-discussed in biology, but is actually quite useful to think about. And I'll illustrate this in a minute or two with some oncology examples. But it's around the idea of domains of validity. This domains of validity is a term that's very commonly used in many fields of science, but as a biologist, I only came across it very late in the day when I was working with a cosmologist who'd become a bioinformatician. And it's the idea that models are predictive within certain sets of criteria or parameters, but they're not predictive within others. And the example that was used with me to explain this is Newtonian mechanics, which is pretty good at explaining the motion of things that are bigger than an atom, smaller than a black hole, and traveling a bit less than the speed of light, right? So if those conditions are satisfied, then Newtonian mechanics does a pretty good job. But if you're smaller than an atom, nearly as big as a black hole, or traveling around the speed of light, then only an idiot would use Newtonian mechanics. And I think as biologists with disease models, we often need to be clearer and also articulate more clearly where those things that our models can predict versus those things that they can't. And if you're interested in this, I've highlighted on the right a paper which is a very rare example of a rigorously derived set of criteria against which you can evaluate models. And this was used by a drug industry consortium to say, here is how we're going to decide whether in vitro models of liver toxicity are good or not. And it divides the territory a bit differently to how I've done it on the left of the slide, but effectively it specifies a bunch of things like, okay, if you've got an in vitro liver model, it should produce a certain amount of albumin, and that should be the same amount of albumin that liver cells produce. It should produce the right amount of urea. It should express a set of genes that we know are expressed in the liver. It should resemble the liver in terms of histology. And also, if we test a bunch of drugs that we know are toxic and close structural analogues of those drugs which are not toxic, it should give us the right answer. And if it does all those things, then we will deem this to be a reasonable model for the purposes of in vitro liver tox, right? It's remarkably how rare that happens. And here's a rare example where it has. And here's an example of an efficacy model, again, from one of my collaborators. And I think, and it's possible one of the authors of this paper is on the call as well. At least she told me she might be. So this is a slightly different framework. Again, a lot of the frameworks are sort of different, have slightly different headings under which they put things. But here are a set of criteria, a checklist where you can describe Duchenne muscular dystrophy in great detail. There would be sort of sub-criteria against each of these criteria. So for example, you look at the genetics or the biochemistry or the epidemiology of the human disease to derive criteria against which you evaluate your model. And then you can look at different models so you can understand their strengths and weaknesses. So in this particular case, it turns out that the dog model of Duchenne muscular dystrophy, this particular dog model, actually in many respects looks a whole lot more like people than does the common mouse model. But actually the common mouse model's had a whole bunch more drugs tested in it, right? Probably because it's cheaper, right? But again, these sorts of plots give you a sense as to where the strengths and weaknesses of different models are. So they help you think about assembling useful sets of models to help you in the decision process. I'm going to wrap up fairly soon, but I'm going to just give one or two concrete examples before I do. On the left are some therapy areas where I know a little, certainly less than some people on the call about many of them. And on the right are some sort of practical lessons, which just illustrate some of the things I've learned when thinking about evaluation of these models. One thing I've learned is that information about models is not merely poor very often in important decision processes, it's often entirely absent, right? So I and some co-authors in a paper that is currently in press, for example, had to review academic grant applications to try and cure particular diseases. I now know that probably the most important thing you want to know before giving the academic money or not is the extent to which the models that they plan to use have predictive validity, right? I've had to review grant applications in the past where there was nothing at all that would give me there's just no information at all about the likely predictive validity of the models that were going to be used to cure the diseases that the academics wanted to cure. Similarly, investigator brochures, right? Doctors being asked to recruit patients into a clinical trial. An extremely important thing you would want to know, in my view, is what is the predictive validity of the models that were used to bring this drug into the clinic. That information is not simply inadequate in most investigator brochures, it's generally entirely lacking. Another thing that's very interesting is that very often models are used and they're asserted to be a model of disease X, when actually there's been remarkably little formal attempt to characterize the clinical state, right? I've been doing some interesting work recently with Alzheimer's experts, and it's clear that the very first step in evaluating the models is actually characterizing in considerable detail what you think Alzheimer's is, because only when you've done that can you say the extent to which the model recapitulates it. And I would say sometimes there's something even worse than the poor characterization of the clinical state. Sometimes, again, John and Ian may have views on this, is you might have what's called the reification fallacy, where you actually confuse the world with the model. So it may be the case, and I'm only suggesting this, that in oncology, for example, the dominance of models associated, for example, with hyperproliferation during the cytotoxic era of oncology drug development effectively encouraged people to think of the disease in real patients as if it were the same as the models, right? So not only can you have poor characterization of the clinical state, which makes R&D inefficient, the models can sometimes force you to think about the disease in people in a way that's wrong. One of my favorites is ischemic stroke drugs around tests and endpoints. There is one ischemic stroke drug that showed efficacy in 19, possibly 17 or maybe 19 different published animal studies. And that drug then went into clinical trials. In the animal studies, the time between inducing the stroke and giving the drug, the median time in those 17 or 19 studies was 5 minutes. In human stroke patients, the median time in the trials was 5 hours, right? Surprise, surprise, the drug that worked in animals when dosed a few minutes after a stroke didn't work in people when dosed 5 hours after the stroke. So I think there's a whole bunch of, so I think when it comes to model evaluation, again, the point I'm trying to make here is very often we're actually starting from a pretty low base. I'm going to just say thank you to a few people and then stop. And so I've seen at least 2 people on this call. Jim Bosley is a ex-engineer who is now into sort of physiological simulations. John is a cancer drug expert. Jerry is a psychopharmacologist. Hubert is a cardiovascular expert. Guy has developed some important frameworks around model evaluation. Duncan Richards worked at GSK. He's now a professor of medicine at Oxford. Mark Traherne's an expert in psychopharmacology, used to work at Pfizer, amongst other places. Jerome's an Alzheimer's expert, as is Pierre. Desiree, again, has done some great work on developing frameworks for model evaluation. Then Lorna, Daniel, Jim, and Demetrius work at a company called Emulate, who I've done some work with, and they've thrown a huge amount of effort at rigorously evaluating some of their physiological systems, so that they actually have data to show that they can do drug-induced liver injury rather than just asserting they can. So I will stop there and see if anyone's got any comments or questions.
Jack, thank you so much. If you could put the slides down so we can see people raising hands. Jim, please go ahead.
Thanks, Jack. As usual, you do a brilliant job of talking about something extremely important and something you're very passionate about. Great talk. You mentioned that we don't often evaluate our models for what they're really supposed to do, and I think there's one aspect of this that I'd just like to touch on. You can comment on, riff on all of these if you want. I have 3 points. I'll be trying to be quick so as not to monopolize. We always use models. Sometimes we use a mental model to integrate lots of complex data, and I've never seen any evaluation of mental models as compared to some of these other things. That's point one. This second point is, models are often thought of as animal models or in vitro models or omics tests or some other assay. They can also be an integration of all relevant scientific information using mechanistic modeling, which is what I do, as you know. I won't tout that, but I will note that as I look down your criteria list, I think we hit all the bullet points, just humbly. The third thing is really a comment on a very often used metric that I've come to realize is not a very good one, and that's probability of technical success. And that's often used in relating how many drugs go into a phase as to what's coming out of it. That's bad because it convolves or conflates false positives and true positives in a very bad way, in a way that changes from when you're early, when you want a low probability of success, to the late stage. But yet in the drug industry, promotions and managerial responsibility and decision-making power is often given to people who have had a high probability of technical success in their programs. Hope that's not too malevolent, but make any comments you want.
Okay, so seeing as we're co-authors, you won't be surprised to hear I agree with a lot of what you've said. So I think I struggle with terminology. I think when I write about this, I use the term decision tool because, which is more general than model. And I think models are an important set of decision tools, but there's a whole bunch of others, for example, the experience one carries in one's head. So I would include mental models. And although I don't know about drug R&D, there is some very nice evidence in the sort of expert judgment literature that suggests that even simple quantitative models tend to outperform experts, provided they're suitably parameterized, right? So I think there's quite a big literature that actually human judgment generally underperforms quantitative algorithms when you can parameterize those quantitative algorithms, even though they're quite simple. I'm going to say something about probability of technical success. Now, apologies for those of you who haven't wasted so much of your life in drug and biotech investment, but a lot of the sort of financial analysis and sort of practical management of drug industry portfolios uses something called probability of technical success. So this is a sort of attrition measure. So people know, well, in general, we have to put 8 things into phase 1, and then half of them will get into phase 2. So the probability of technical success is 0.5, and then 1.5 of them get into phase 1, so the probability of technical success there is roughly 0.3. Now, my view, for what it's worth, is that that's a very dumb way of building your analysis pipelines, because probability of technical success, as Jim says, conflates good and bad decisions, right? So if we have attrition, it could be because we're throwing away good drug candidates, or it could be because we're throwing away bad ones. And actually, there's a huge difference between the two, right? And again, I won't go into the details here, but one of the areas that we've been actively doing some work in is to develop better methods for valuing pipelines, so that you can actually plug in assumptions about decision quality. And if you do that, it suddenly says, if you get a slightly better model, it's worth $100 million. So by doing that, it actually encourages people to invest in models. right? So you've got this funny situation where at the moment, the financial tools that people use to evaluate pipelines are blind to model quality. If you made them unblind to model quality, maybe a bit more capital would flow into better models because people could see easily how much better models were worth.
Thank you, Jack. Any other questions, comments? Jack, I have a question for you. Not understanding the field very well in terms of approach to drug discovery, whether it's in academia or it's in commercial sector. Is it all about models, or other techniques being used? And for example, now with increased capacity, there is ability to test millions and millions of samples with different reagents just to see what works out. This is trial and error kind of approach rather than modeling approach. So what is the prevalence of modeling prior to the next step?
Okay, so again, I use models in a sort of very broad sense, and again, perhaps decision tool is a better term, right? So I think in pretty much every, well, so if you think about sort of conventional bottom-up drug R&D, right, you start off with a large set of therapeutic possibilities, and those might be targets, i.e. bits of biology against which you intervene, or they might be, you might know which bit of biology against which you intervene, but you're looking for chemicals that intervene effectively against that bit of biology. And I would regard anything that anyone uses to try and sort of narrow down that set of possible things into one or two things that are more likely to work, I would regard all of those as decision tools. Right now, some of them are formal screening and disease models, right? So some of them are in vitro tests, like does compound X bind protein Y in a high throughput screen? And then some of them will be much later on, you know, for example, does compound X improve Parkinson's symptoms in a primate model of Parkinson's, right? But again, I think the logic, the sort of across a very broad range of that spectrum, the decision theory suggests quality beats quantity, right? So testing lots and lots of things in a model that's not very highly correlated with the human outcome of interest is generally a bad way to go. And I'll give you another interesting example. There's only about 1,500 approved drugs or approved drug compounds. It's not many. So the whole pharmacopoeia that is used in people is actually very constrained. It's a very, very small set of chemistry. And chemical space is almost infinitely large. Now, if you look at the uses of, if you look at the new uses of drugs, and indeed so if you look at drugs that are launched, and then you say, well, how many subsequent indications or subsequent respectable uses will those drugs get? What you find is an extraordinarily large proportion of those uses come from field discovery. Right, so drugs are launched, then 20 years later you say, well, what are these used for? You'll find out they were used for a bunch of things that they weren't initially launched for. And most of those things, at least in the relatively small studies that have been done of this, the uses were discovered not by scientists, but by users, right? And my view is that's quite an interesting illustration, because if you're using real chemicals in real people, your throughput is very low. It's not like you're testing lots and lots of compounds, because there's only 1,500 approved drugs, right? But you're discovering uses. And the reason for that is real people with disease X are a very good model of real people with disease X, right? So if you know, so I think field discovery is another, or the importance of field discovery is another way of showing that quality beats quantity, that humans with a disease are of a very high predictive validity model of other humans with the same disease, which means with very small numbers of humans and very small numbers of drugs, we can still discover quite a lot of useful things.
Okay, thank you. Andrea, your question?
Yeah, hi. I very much like your approach of the sort of checklist to try to come up with more predictive models. at the end of the day, though, I guess really it's only the predictive validity that matters. And these other things are basically, hopefully, proxies of predictive validity.
Yeah.
But it's ultimately that test of predictive validity is in a way all that matters. Thinking of your liver tox example, if the model doesn't express as much albumin as you might like, but it turns out to be more predictive, of course, it's going to be the better model. And so I wonder if there's a way to deal with that, and then also maybe if you could just give an example of a model that you think meets most of your criteria but turned out not to be good.
Okay. So I think you're absolutely right. So those checklists, or actually most of it, in diseases where there's been a large number of compounds have gone through models and into people, right? Or in tox in particular, because tox is actually quite rich data for doing this sort of work with, because unlike many efficacy models, everything has to go through tox models. So you actually have much more data to play with, and you can actually go and get compounds and test them all in your tox model, and you at least know to an extent how toxic they are in people. So tox models are arguably the easiest to do this in. I think it'd be really nice to do, I haven't done it, is to get a bunch of tox models that don't actually recapitulate the human biology and see if they're worse at tox. Right, so, and again, I can talk a bit about the work I've done with Emulate, because they've published it, right? So it's in the public domain. But, they got into what they call microphysiological systems or organs on chips, because their view is that the phenotype of lots of cell cultures just stops being like the phenotype of the tissue of interest after a while. So sure, they're liver cells, but once you put them in a vat and grown them for a while, they stop behaving like liver cells, right? They're not liver cells anymore. But I think with other diseases, maybe type 2 diabetes models, you could also do, there's enough drugs that go into people. I think in some oncology models, but very often there won't be. But again, I think the important thing, or the thing that strikes me, is what a low base we're starting from, right? It's not like actually a lot of these people have done really good checklists on these models. I think lots of models there's been almost no formal evaluation at all. And people use things because of availability bias. It's because of what they've always used, or because what it's easier, it's the cheapest thing to use, right? And so again, I come back to my Bayesian point. The changing the correlation between your model and the human outcome of interest by a factor of 0.1 can have the same productivity impact as doing 10 or 100 times as much stuff, right? So we don't have to, your checklist doesn't have to nudge stuff very far in the right direction or give you a particularly great insight into which of the model's better or worse to probably be quite useful, right? And something I've proposed, and no one's done it yet, is that, for example, one could do sort of historical studies to back test stuff, right? So a bit like they do in machine learning, right? You divide the back catalogue of several large drug companies, you have several hypotheses about how you can characterize models. You take half the data, you assess the models in that half of the data and see whether they predict things better or worse. And then you look at some untested other models and see if you can still do it, right? And that might be a way of doing better evaluation frameworks. So there's ways of doing it, but I think it would be quite a lot of work. Your second question, so things that have been evaluated look really good that have been poor. Really interesting. I haven't looked for that. I've looked for things that have been poor and then sort of retrospectively tried to understand why they were poor, right? I've looked for things that have been good and then tried to retrospectively understand why they're good. And I would love to do that, but I haven't done it yet. And if anyone's got bright ideas, let me know. Because in a sense, that'd be good. The surprises are interesting, right? So it would be good to find the surprise. Now, there are some models that there's no particular reason why I think anyone believes they should work, but they do. And again, I may be showing my ignorance here, but for example, there's really obscure things like, for example, there are certain guinea pigs or certain rodents are very good at detecting the anxiolytic effect of benzodiazepines, but only benzodiazepines, right? Why that should be the case, I've got no idea, right? So I can think of good ones that no one understands, but I can't think of things that should be good that are bad.
Thanks, excellent talk.
Okay,
Can I ask
Jim,
an-
your
Can I
comment?
ask another question? If I can ask one question. This highlights the problem of false negatives and assessing the number of false negatives. Care to comment on that, Jack?
So I didn't. So Jim, I'm going to plug the paper that you and I published in 2016, where we talk about this in a bit more detail. But I think there's a sort of assumption that we don't throw away good stuff, i.e., the drug industry thinks in terms of attrition. We're trying to get rid of all that bad stuff. But actually, if models just are poorly correlated with reality, you're not just progressing bad stuff, you're actually throwing away an awful lot of good stuff, which comes back to this point about field observation, right? So why is it that so many uses of drugs are observed in the field? It's because you've got a high true positive rate, right? It's when you have poor models, when you've got very high throughput and a poor model, you're throwing away the vast majority of good drugs, right? So actually, if you've got a good model and high throughput, and a bad model and high throughput, you can be pretty sure that nearly all of the good drugs are rejected, right? And again, our 2016 paper talks about that in a bit more detail, about how it's not simply progressing good stuff, bad stuff. You throw away lots and lots of good stuff with bad models, or rather, you have a very low discovery yield, right?
Yeah.
All right, thank you so much. So we're running out of time, but if there are any final comments, questions, it's an opportunity to raise your hand now. All right, so Jack, thank you so much once again for educating us.
Well, no, thank you very much for the opportunity. It's a lovely group to present to, so thank you.